So what's the big deal about oxygen anyways? I mean, we talk about it, it's all important, we're always worried about it. And funny thing about it is oxygen lives in this world where it's both necessary and deadly. I mean, 4 minutes without it and you're dead. But if you breathe pure oxygen, it'll burn your lungs. So today we're going to explore what it is about oxygen that makes it such a big freaking deal. Now, I'm sure you think that, oxygen's, it's obvious. I mean, you use it in combustion and it's used to make ATP. But I bet you don't really understand how that actually happens. So today, let's take a trip and see. The first thing we need to do is get to the cells. The fundamental problem that all metazoans have to solve is how to get oxygen to the cells to do the work that it needs to do. And in order to get oxygen to the cells, there's a number of different barriers it has to work through. And now the first barrier is the alveoli. How much oxygen gets through the alveolar membrane is determined by a fixed law. It may never have occurred to you, but unlike many other important molecules in your body, there is no active transport system for oxygen. All of this happens by diffusion, and Fick's law is the law that governs diffusion. It is the ratio of the permeability of oxygen within its medium, the surface area available for diffusion, the pressure gradient, and also the distance for diffusion. Now, Fick's law in of itself is not important to remember, but one of the most important critical care formulas that you need to understand is derived from it. The PF ratio is the ratio of oxygen that is dissolved in the blood compared to the ratio of oxygen that's in the alveoli. This is derived from the Fick's equation using the difference in pressure across the alveolar membrane. Now, an easy solution for any organism would be simply to create a system of tubing with fluid and just dissolve the oxygen. And in fact, many metazoans that are small will use this as an option. Basically dissolve oxygen and then just pump it around. But as it turns out, free oxygen in the blood is actually a pretty poor way to transport oxygen. Probably the biggest issue is solubility. And one of the big factors that determines solubility is the temperature of the liquid. Now, if you've ever seen water boil, you can intuitively understand that as the temperature of a liquid goes up, the solubility of the gas dissolved in it goes down. Now, maybe solubility is less of a problem for cold-blooded creatures because they can dissolve more oxygen in their blood because they're colder, but for those of us who are warm-blooded, it's a bit of an issue. And it even becomes more so when you're running or exerting yourself or trying to chase down that prey or run away from being prey. In addition to temperature, partial pressure of gas also determines solubility. So now in a human at 35 degrees Celsius, 0.03 milliliters of oxygen can dissolve per liter of blood for each millimeter of mercury of partial pressure of oxygen. So if you calculate this out at a PaO2 of 90, you get 2.7 milliliters multiplied by 5 liters of blood, and you get about 11.5 milliliters of oxygen that can be dissolved in the total body blood volume. Now, if you give somebody 100% oxygen, you push the PaO2 up to about 400, and you get a total of 60 milliliters of oxygen dissolved in the blood. So in exercise physiology, the concept that is commonly used as a measure of exertion is 1 MET, which is approximately 3.5 milliliters of oxygen consumed per minute per kilogram. So for the average person, you would consume about 262.5 milliliters of oxygen per minute. So you can see that this is a bit of a problem. Not only can you not afford to ever hold your breath, you probably also shouldn't be running. Now, an alternative way to transport important things is to bind it to protein. In fact, many important molecules are bound to protein in the blood in order to transport them. And in the case of oxygen, hemoglobin does that job. Hemoglobin is such an awesome molecule for transporting oxygen. There are literally millions of organisms that have evolved a hemoglobin molecule or a hemoglobin-like molecule to help it transport oxygen. In a human being, you get an astounding amount of oxygen transported with very little extra work because of these molecules. So let's take, for example, a hemoglobin in a normal person of 150 grams per liter. This means that the total amount of hemoglobin in the blood is about 750 grams, and this is capable of transporting over a liter of oxygen at a time, because 1 gram of hemoglobin can bind 1.34 milliliters of oxygen. So an incredible gain in the ability to transport oxygen. So if you're an organism trying to decide how to transport oxygen, dissolve it or bind it, you can see that it's a bit of a no-brainer. I mean, 262 milliliters of oxygen consumed per minute, it's really simple. Probably should bind it. This curve is the relationship between the amount of oxygen that can be bound to hemoglobin and saturated relative to the amount of the partial pressure of oxygen in the system. You should notice that the curve is a sigmoid because each molecule of oxygen that binds to hemoglobin changes the conformation of the other hemoglobin molecules in the tetramer, and it changes the binding affinity for the next oxygen molecule to bind. This is a concept called cooperativity. On the curve, the key point is what we call the P50 mark. This is the PO2 needed in order to saturate 50% of the hemoglobin. This can be used as an anchoring point when you're talking about the shifting of the curve to the left or the right, because the environment that the molecules exist in alters its binding capacity to oxygen. When you shift the curve to the left, the P50 decreases. This means that the PO2 necessary in order to saturate 50% of the hemoglobin molecules decreases. Conversely, if you shift the curve to the right, the P50 increases, which means that you need a higher PO2 in order to saturate 50% of the hemoglobin. Now, a common question that will appear on exams is for you to name conditions that cause a left or a right shift. Things that cause a left shift include hypothermia, alkalosis, and carbon monoxide. On the flip side, hyperthermia, acidosis, and carbon dioxide are things that cause the curve to shift to the right. I find that it is easy to remember what things cause the curve to shift to the left or the right by just trying to remember at what points when I want oxygen to be more avidly bound and when would I want it to be released. Well, I would want oxygen out of the hemoglobin when it is in near the cells, and that's an environment that is warm, acidotic, and there's lots of carbon dioxide around. Whereas you want the oxygen to be bound more avidly in a place where it should be picked up quickly, and that would be the lungs. And the lungs are cold, there's not a lot of carbon dioxide, it's an alkalotic environment, and this is also the place where you get exposed to carbon monoxide. So that's the little mnemonic way I remember which way the curve shifts and under what circumstances the shift occurs. This shifting back and forth occurs in the lungs and the tissue normally, and this is called the Bohr effect. Now, there may be 50 ways to leave your lover, but only 2 ways to transport oxygen. And this actually makes things easier because you can then calculate the total amount of oxygen that can be carried by calculating the oxygen content of blood. The oxygen content of blood equation is a fundamental equation in critical care medicine. You must know this. And because there's only 2 ways oxygen can be transported, calculating is straightforward. The first term is the amount that can be carried by the hemoglobin molecule, and that's 1.34 multiplied by the hemoglobin concentration and the oxygen saturation. The second term is the amount that's dissolved in the blood, and that's the 0.03 multiplied by the partial pressure of oxygen. Now, while it may seem a bit pedantic, knowing the actual units matters. The oxygen content is reported as milliliters of oxygen per liter of blood. And this matters because in some labs, the hemoglobin portion is measured differently. Standard should be grams of hemoglobin per liter of blood. But if the lab uses a different unit, such as milligrams per deciliter, then the term 1.34 won't fit, because that term is 1.34 milliliters of oxygen dissolved per gram of hemoglobin. And so the terms won't cancel out. So it's important that you know the individual units so that you come to a final units of milliliters of oxygen per liter of blood. Once you know the oxygen content of blood, you can calculate the total oxygen delivery by multiplying the oxygen content by the cardiac output. And you can see with the terms how it cancels out, so that liters cancels out and you're left with milliliters of oxygen per minute. Now, if we assume a cardiac output of about 5 liters per minute, which is resting for a normal person, and a hemoglobin concentration of about 150 grams per liter, the calculation is pretty straightforward, and a normal oxygen delivery is about 1 liter of oxygen per minute. Now, we've been talking about oxygen and assuming that this is on the arterial side, but if you take it on the flip side and calculate the amount of oxygen that is in the venous system, you can use that information to calculate the oxygen consumption. The normal amount of oxygen consumption is simply the difference between the amount of oxygen that's in the arterial side minus the amount of oxygen that's left over on the venous side. If you calculate those two oxygen contents and multiply it by the cardiac output, you get the amount of oxygen consumed, or the VO2. Normal consumption is about 250 milliliters of oxygen per minute. Now that we know the equations for calculating the oxygen consumption, delivery, and also the content, let's have some fun and let's see if we can figure out what the required cardiac output would be in order to support an oxygen consumption of 250 milliliters per minute without hemoglobin. So the equation is thus, where we're going to, in this case, exclude the terms for that amount of oxygen bound to hemoglobin, because we're excluding it. Now isolate the cardiac output, and we'll have the cardiac oxygen consumption on top of 250 milliliters per minute, divided by the amount of oxygen consumed, and this is 0.03 multiplied by the PaO2, which is about 90, minus 0.03 times the PVO2, which is about 40. And this leaves us with 0.03 multiplied by 50 gives you a cardiac output of 166 liters per minute. So since we only can generate 5 liters a minute on a normal day, you can see why it's important that all metazoans have some form of an oxygen carrier, because there is no efficient way to support high metabolic demands using oxygen just dissolved in the blood. One other equation that's important to know is the extraction ratio. When you know the amount of oxygen in the blood leaving the lungs and returning to the lungs, you can calculate the extraction ratio, which is a measure of the amount of oxygen that's been taken up by the cells. This is the difference between the oxygen content on the arterial side minus venous oxygen content divided by the arterial content of oxygen. I'm not going to belabor the point. You can cancel out a lot of the terms in the calculation except for the arterial and the venous saturations, and for the most part you can ignore the amount of oxygen that's dissolved in the blood. This leaves you with the difference between the saturation of oxygen as it leaves and minus it from the oxygen saturations as it returns to the lungs, and divided by the oxygen saturations on the arterial side. A normal extraction ratio is about 25%. A high extraction ratio means that the cells are starving and are taking up as much oxygen as possible. An example of this would be in cardiogenic shock. Now, on the flip side, a low extraction ratio is caused by intercellular shunting, where blood is not getting to the cells that need it. And you'll see this oftentimes in sepsis. You can use this calculation as a rough guide if somebody has a central venous catheter in place, but ideally this calculation is done best when a Swan-Ganz catheter is in place. Now, one last concept we need to cover before we get out of the lungs is we need to talk about the blood that misses out on the gas exchange experience. There is some blood that will bypass the alveoli. This is a shunt, and this can either occur at the heart level from a right to left shunt, or it can go past flooded alveoli, as you would see with consolidation, atelectasis, pulmonary edema, or severe pneumonias. The key point to understand is that the amount of oxygen in the bypassing blood doesn't change. This means the shunt ratio can be calculated as the difference between the end capillary oxygen content minus the arterial oxygen content divided by the end capillary oxygen concentration minus the venous oxygen content. The end pulmonary capillary oxygen content is this equation, which is not initially intuitive, but you think about it as essentially the theoretical maximum amount of oxygen content in the blood in an ideal lung. It's the amount of oxygen at 100% FiO2 plus the total amount of oxygen that's in the alveoli. And this equation, the PaO2, is a byproduct of the FiO2 multiplied by the partial pressure of minus the PaCO2 divided by the respiratory quotient. Much like when you're calculating the AA gradient, you use the respiratory quotient and you can assume it to be about 0.8 in most patients, just as a rule of thumb. Now, riding through the blood is an exciting ride, but it's time for this oxygen to get to work. So the next stop is the mitochondria. The mitochondria, interestingly, evolved from archaebacteria, and their relationship with eukaryotes is basically shelter for energy. And there's also some conjecture that these bacteria may have also played a role in protecting the cell against the toxic oxygen environment that was the earlier Earth. Now, as a general concept, you have to understand that only organic molecules burn. Burning essentially releases a lot of energy at once, but mostly as heat, not very useful for a cell. These carbon-carbon covalent bonds have their electrons at higher energy states, and when the bond breaks, the electrons flow to a lower energy state and release that energy. Organisms can take advantage of the electron flow energy release to generate ATP, but they have to be able to control this reaction. Glycolysis and the citric acid cycle progressively break down glucose to capture the energy of the covalent bonds in the glucose molecules. Generating ATP directly from this is not as efficient as more energy is released in the bond than is used to make ATP. So instead, the molecule NADH is used to capture most of that energy. The Gibbs equation is the fundamental equation for thermodynamics. This is the amount of Gibbs free energy is equal to the amount of enthalpy minus the temperature and the entropy. All spontaneous chemical reactions have a negative Gibbs free energy. You can see from the equation that it's easy to generate a negative Gibbs free energy if you use a high enough temperature or if the substance has a very high entropy. Creating ATP from ADP and phosphate is a chemical reaction that requires a positive Gibbs free energy. So in order to generate that type of a reaction, you need to pair it with a negative Gibbs reaction. Now, back in biochemistry, you probably were told that the glycolysis makes 2 molecules of ATP, whereas the Krebs cycle makes 38. But you probably also noticed that the cycle itself didn't show that 38 molecules of ATP being made. So where's this ATP? I mean, they say it's important, and then they don't actually show it to you. And there's all this NADH and FADH. Like, these biochemists are out of their mind. But if you mix NADH or FADH with oxygen, you get a very negative free energy release. The chemical reaction is NADH plus oxygen yields NAD plus a proton plus an electron plus an awful lot of extra energy. So now you can use this energy from the oxidation of NADH and oxygen to be used to make ATP. But phosphorylation of ADP to ATP requires about 57 kilojoules per mole. But the actual reaction of NADH plus oxygen generates negative 158.2 kilojoules per mole. So there's a lot of excess energy for the chemical reaction. So this is a little inefficient and probably a waste of energy because there's no way to capture it. But if you want to capture this energy efficiently, you need to use a transmission in order to control the transfer of energy. You see this every day when you're driving a car. For example, an engine will run at about 2,000 RPM. The purpose of the transmission is to stop the wheels from going that fast. If you took the speed of the engine and directly transferred it to the wheels themselves. The electron transport chain in the mitochondria is the biological transmission. It passes electrons from the reduction of NADH and oxygen to fuel the pumping of protons out of the inner membrane. So now, outside the inner membrane, there are a whole bunch of protons that have been pushed against their concentration gradient. And the mitochondria then uses the F0, F1 ATPase to phosphorylate ADP into ATP using the energy associated with the protons flowing back down their concentration gradient. Well, now you think, gosh, that's really cool biochemistry, Dr. Hudson, and I've now learned so much more than I knew even a mere hour ago. Clearly, you know many things, but none of the things that I've shown you in terms of the Krebs cycle actually involves oxygen. So what's the deal with the oxygen? Oxygen's actual only job is to be the final electron acceptor in the electron transport chain. The electrons are passed on to the oxygen molecule and in combination with some proton is converted into water. And now, as a bonus, you can also see why chemicals like carbon monoxide and cyanide are so toxic. These chemicals block cellular respiration by blocking oxygen out as the final electron acceptor. If there's no oxygen or oxygen can't take its place in the electron transport chain, the entire chemical reaction stops. So there you have it. Oxygen is a dichotomous molecule. You need it in order to live, but it has a relatively simple role in cellular respiration. But beyond that, and you need to understand some very important things about oxygen. You need to be able to describe the PF ratio. You need to appreciate how oxygen delivery occurs in the body. You should be able to describe the equation for the oxygen content of blood and be able to derive oxygen delivery, consumption, and the extraction ratio, as well as calculating the shunt fraction. And then finally, you should be able to appreciate the use of oxygen in the mitochondria. Thanks for watching. Until next time.